Okay, good afternoon. I hope all of you have a good experience, had a good experience on the exam, quiz exam, and this is your first quiz exam with me, and I hope you enjoyed it. Today we shall briefly discuss the concept of gene editing. Do you have any idea regarding the gene editing? What is gene editing? Do anyone have any idea regarding gene editing?
Yes, sir.
Yes, please tell me, what is gene editing?
Gene editing is a branch of science in which a certain segment of gene is selected and change in a desired way. It can be removed or changed or edited.
Excellent. Do anyone have anything to add?
Sir, it mainly is done is to get the desirable one and to remove the undesirable one.
Great. Let me tell you in details of gene editing. Yes, gene editing. There are. Can you see the slide?
Yes, sir.
Okay,
Yes, sir.
excellent. Gene editing is a new revolutionary technology in biology. We can call it bioediting also. And among the gene editing, the CRISPR-Cas9 gene editing or genome editing technology is very important because it is making a revolution in our agriculture also. Not only in agriculture, it is applicable to any branch of biology. And in agriculture, you know, due to climate change, agriculture is now in a very big challenge to ensure the food security of the growing population of the world. I discussed in my earlier lecture that the world population will be increased to nearly 10 billion by the year 2050. And to feed the additional nearly 3 billion population, we need to increase our food production 70 to 100% from now. And climate change is posing a serious threat to increase the food production. So how can we make climate smart or climate resilient agriculture? It is a big question. And big blessing is now associated with the CRISPR-Cas genome editing. Today, I shall focus CRISPR-Cas genome editing with, also brief discussion on other genome editing or gene editing technologies. And now let us know what is gene editing or genome editing. Precisely altering genetic sequences in living cells, including those of humans, at much higher accuracy and efficiency than ever before possible. And this definition was given by David Baltimore, a Nobel Prize winner in 1975. And gene editing can easily be defined as any techniques using site-directed mutagenesis to make small changes to DNA sequence at a known location is called gene editing or genome editing. That means site-directed. In the DNA sequence, very specific target you can fix, and for example, to change a bad gene, you can target it and you can make a mutation in that gene and or knock out it, and then you can make it dysfunctional. As a result, you can improve the trait of the organism. So gene editing is a side-directed mutagenesis which can lead to the improvement of the organism. Targeted mutagenesis or genome editing is changing the genome at a specific site, your desired site. Changes can be two possibilities, two ways you can do the changes. One is small deletion. Small deletion means ATGC sequence, you change one or two, nucleotide, somehow delete, and then it result in non-functional phenotype of the particular gene. So changing effects of specific gene, usually knocking it out. Change a DNA with another piece of DNA, desirable gene variant, that means allele. That means you can change a small part of DNA by using another small part. So you can introduce the DNA. So you can do the change in the genome by two ways. One is deletion, another is addition. In case of addition, the product would be transgenic. In case of deletion, the product is just a mutant. No transgene is present there. As a result, it is not necessary in many countries for biosafety guidelines. So how can we do it? DNA repair mechanism. If somehow by UV ray or any other mutagenic agent, if you make a lesion or cut the double-stranded DNA, then what would happen? It immediately repair. And during this, repairing mechanism, that base excision or nucleotide excision taken place. That means some nucleotide cut and removed from the DNA sequence. And if you make the cut in your desired location, then you can knock out a gene or make a non-functional gene, because DNA has the inherent mechanism of repairing any injury. So by using some, technique, here you can see the scissor. Scissor means some enzyme. You can cut the DNA, and then DNA immediately repair itself, leaving some of the nucleotides. So the gene present there becomes non-functional, and it is a mutant, and it is safe, and it would carry a good trait, because if bad trait is removed by this way, then the outcome would be a good trait. And it can be done two ways. If you cut the double strand by using specific, agent, for example, site-directed nuclease. Nuclease means that cut the nucleic acid. Then non-homologous end joining. Automatically, the two end can be joined, leaving some of the nucleotide cut and outside, that is nucleotide excision mechanism, and a mutation taken place. But if you cut a certain DNA in a certain location, and if you put a template of DNA which you want to add here, then that DNA template can be immediately added here, and then ligase enzyme, repair paste it, and then you would get a new genome, genome with a new gene that you wanted to introduce in the certain location. So there are two ways of DNA, a natural repair mechanism. One is automatically mutation, and another is, to introduce a new specific gene or sequence, so that you can make changes in the genome of an organism. Both is genetic engineering, both is genetically modified organism, but here no transgene, and in homologous recombination there is a transgene. Both can be done by using now modern gene editing technology. So targeted DNA cutting enzymes are called nuclease. If you are asked what is nuclease, nuclease is a DNA cutting enzyme. So DNA you cannot see, you cannot use the scissor. So scissor of cutting DNA is nuclease. Recognizing specific site in genome by a DNA binding domain. So you can add a DNA binding domain so that it can recognize a specific site of the DNA which you desired. And there is a cutting domain which cut, and site specific nucleases means that nucleases enzyme can specifically target a specific site, or it can be called site directed nucleases. So you can direct it to go there. How you can do that we shall discuss. Programmable nucleases or designer nucleases. So there are several, all are synonymous. One method of gene editing using the nuclease cut the DNA is zinc finger nucleases, which was discovered in 2005. And another one is TALENs, transcription activator like effector nucleases, discovered in 2009. And the latest one, CRISPR-Cas, which was discovered by the scientist in 2012. CRISPR stands for clustered regularly interspaced short palindromic repeats, and Cas is an enzyme associated with the CRISPR, and scientists discovered CRISPR in prokaryotic organism like bacteria and archaea. Now let me tell you briefly about zinc finger nuclease. Zinc finger nuclease derived from family of transcription factors. Transcription factors means factors involved in transcription. Transcription means from DNA to RNA, in the central dogma. Recognizing specific sequence in gene promoters. Gene promoters, they can specifically recognize. Peptide units of which the 3D structure is stabilized by zinc ions, leading to substructures looking like fingers. The substructure of the peptide which look like finger, this is why finger name is comes, and zinc ion is involved there. This is why zinc finger and nuclease. Fingers can be assembled to recognize a specific 3-base pair DNA sequence. An array of these can target a longer sequence in the genome. For cutting this target sequence, a nuclease called FOCI, is added to the ZNF. So a nuclease is added to the designed, nucleases, guided nucleases in the zinc finger nuclease. And the enzyme FOCI, naturally found in Flavobacterium, is a bacterial type 2S restriction endonuclease consisting of an N-terminal DNA binding domain and a non-specific DNA cleavage domain at the C-terminal. So it is already discovered from the bacteria, the enzyme FOCI, and it is associated, it is the real nuclease involved in zinc finger nuclease. So zinc means the zinc ion, finger means the peptide structure like a finger, and nuclease is the FOCI used in the ZFN. And ZFN, after discovery, several plants' genome have been, edited. But this technology has some, cumbersome, that is very difficult, not so, user-friendly like your mobile phone. So FOCI nuclease is only active as dimer. So ZFNs are applied as pairs. Doubles the specificity for DNA binding domain. It doubles the specificity. So this is the about the first discovered gene editing technology. We are not going more details at your stage because it is enough for your stage, but we shall discuss a little bit why ZFN are no more longer used by the scientists because of new technology, gene editing technology, CRISPR-Cas is being developed, and it is much more user friendly. I told you that food security is a big challenge, and it is a very challenging task. Already I discussed in my earlier part of the lecture. So you can go through in detail. We have to increase the food production significantly, 100 to 110 times in some scientists estimated to feed the global population. And these are the some summary of the reasons where food security ensuring is a challenging task globally as well as in Bangladesh. Another important information associated with the food security is the United Nations organization set sustainable development goals for all the countries. 17 goals are set, which have to be achieved by the year 2030. Among them, number 1 is end poverty for all. Number 2, freedom from hunger, and others, sustainable, sustainable development, sustainable consumption of and production. These are very closely related to our agriculture, and we have to achieve it. To ensure climate resilient agriculture, we need to produce a drought, salinity, extreme temperature, heavy metal toxicity, short duration crop, biofortification, that is nutrient enriched crop, high yielding crop, and domestication of the wild relatives or orphan crops, because the crops all over the world, we are using only less than 20 crops as our food, but more than 400,000 plant species are still unexplored, not yet been used as in the breeding program for developing the new variety. And in plant breeding, in last couple of centuries, the age of agriculture is about 10 to 12,000 years. So human over the years domesticated plant from the wild progeny, and lots of big discoveries in scientific arena, like Darwin's evolutionary theory, Mendel's Gregor Johann Mendel's law, and many other scientists, they contributed over the years to enhance the ability of humans to increase the improve the quality of the our crop plants. And the recent one is the gene editing, recombinant DNA technology. I discussed you a lot, and this is the chronology you may see later on. But CRISPR is the latest one. CRISPR, already I told you what it stands for. It is a genome editing technique that targets a specific section of DNA, makes a piece, a cut break at the target site, and it has it can do one of two things. What are those? Makes a gene non-functional and replace one version of gene with another. And these four guys simultaneously discovered the CRISPR. They have been considered the promising candidate for Nobel Prize for this discovery, but it may take a few years to get such a recognition. So you can imagine that CRISPR is incredibly powerful technology. This guy from the Lithuania and this lady Jennifer from the Berkeley in USA, Emmanuel from Europe, and this guy Feng Zhang from MIT. And they simultaneously discovered CRISPR in 2012. But CRISPR was first introduced this technology for editing of plant genome in 2013. And one of our collaborators, Sufyan Kamaun, first did that job. And now I would like to tell you a bit background of the discovery of CRISPR. Here you can see the bacterial cell. Bacterial cell is, like human, bacteria also are attacked by the virus. So make the bacteriophage or like this, because virus, they cannot replicate themselves. Virus are either DNA or RNA, particle coated with the protein. So virus needs, bacteria or other host for replicating their DNA or RNA. So virus always infect the bacteria, and over the years of evolutionary process, the bacteria, cut small pieces of DNA from the virus and added to the genome of the bacteria. And this is repeatedly found in the bacterial genome. And they also developed over the process the, that CRISPR associated proteins, so that they can be, how to say, do transcription and translation protein, and then they can use the Cas enzyme, CRISPR associated enzyme, to newly invaded to tackle the newly invaded virus, to cut the DNA of the virus, and then virus cannot, use the machinery of transcription and translation machinery of the bacterial DNA. As a result, a virus failed to infect the host bacterium. And in 1987, Ishino from Kobe University, he first found this type of strange thing. In the bacterial DNA, he found that repeated presence of the sequence. So it is, how it was evolved. First, when virus infect the bacteria, bacteria recognize the viral DNA and then use CRISPR-Cas scissor, molecular scissor, for cutting the viral DNA and show the immunity. So it is one kind of immunity, one kind of molecular scissor that bacteria and prokaryotic, archaea use to stop, invading or infecting virus. And over the years, from the 1987 till 2012, scientists confirmed that the CRISPR strange, repeated sequence, palindromic sequence in the viral genome is nothing but it is a molecular scissor associated with the immunity of the bacteria. Then scientists made a revolutionary thinking. Can we use this, scissor for the editing of any organism's genome? And that was the big discovery made in 2012. And it was possible at that time to prove that if we design the guide RNA with the CRISPR-Cas enzyme, and then we can target any specific sequence in any organism to cut it. So cutting the DNA, and then DNA has the ability to repair it, and then if it is repaired, then mutation taken place. And it was the first starting, point of gene editing by the, Jennifer, Emmanuel, Feng Zhang, and the, other scientists from the Lithuania. So CRISPR-Cas genome editing timeline from the 1987 to if you see 2017, how it is moving now 2020, we have now prime editing technology, more precise, without any off target. We can use the CRISPR-Cas gene editing for the editing of the genome of any organism. So this is the timeline and how it works. Here you can see CRISPR is the sequence, and we can design a guide RNA complementary to the target DNA sequence you are specifically want to cut. And then that guide RNA with the CRISPR-Cas comes to the cell and find the specific location and make a double strand break. And there are several enzymes. Cas9 is one enzyme, CPF1 or Cas12a is another enzyme. There are several enzymes now scientists discovered from the microorganism like prokaryotes, and they can be used. So when double strand break, non-homologous end joining, I told you immediately repair, so gene deletion taken place. Indel means just one nucleotide is somehow removed. Here a big gene can be deleted, or gene insertion can be taken place, or if you keep a template can be inserted. So these are the way CRISPR-Cas principle is worked. So various kinds of genome editing and manipulation by CRISPR-Cas system. Here illustration, the simplest illustration I tried to show you, and it is drawn in our laboratory, and I am showing you so that you can easily understand. So what is unique in CRISPR-Cas technique? Why CRISPR-Cas technique is so superior? Derived from a natural process, that is from bacteria and archaea. We discovered from a natural process and natural immune system found in bacteria to protect themselves from the pathogen. Pathogen means virus. Targets a gene for editing or regulating. Comparable to Photoshop. This is a very powerful word. In Photoshop, I am not sure how many of you are expert in using Photoshop. So like Photoshop, you can edit the DNA in any place. Either you can delete a gene, you can knock out a gene, or you can make non-functional. Even you can make a change in the single base called base editing, and then you change a property that is phenotype of an organism, and you can add a new gene also. The future of CRISPR-Cas, this future predicted by Jennifer Doudna in their paper published in 2014, the future CRISPR-Cas9 mediated genome editing. I can tell you again, now not only Cas9, there are several Cas enzymes have been developed. So Cas9 is popularly being used, Cas12a also popularly being used. So you can do human gene therapy, so specific gene, for example, diabetic or cancer associated gene, which is lethal for the human, you can do gene therapy, you can correct it. Screen for drug target internally, and ecological vector control, mosquito sterilization, viral gene disruption, pathogen disruption. For example, we can edit the gene of the coronavirus also by using CRISPR-Cas. And in agriculture, we can improve crops, animals, and fishes by editing of the genome, their genome by CRISPR-Cas technology. So in synthetic biology, you can make changes in the DNA sequence of any organism as you like, and you can make huge biodiversity with variable traits of an organism. For example, a rice variety, Breedan 29, if you use CRISPR and chop different places in the genome, you can make huge diversity of the rice. And programmable RNA targeting. That means you can design RNA for a specific target of the DNA. Even RNA editing can be done by using CRISPR-Cas. And CRISPR-Cas can be used in biology, in biotechnology, in biomedicine. So it is, amazingly been progressing. You may ask a question, are there evidence that CRISPR-Cas has been used in human and other animals, including human, in almost all the, economically important organisms or model organisms, CRISPR-Cas technology have successfully been used. So it is now a proven technology. So CRISPR-Cas technology is superior to the ZFN and TALENs because other two technologies are cumbersome. Cumbersome means very complicated, requires sophisticated protein digestion, synthesis, and validation. But CRISPR-Cas, as I mentioned, cheap, relies on guide RNA to target site specificity, can be readily designed based on genome choices, and in multiplex fashion. Multiplex fashion means in a DNA, different places at a time you can make editing, not a single place only, which several genomic loci can be perturbed or edited at the same time. System create stable and heritable mutation. This is very important. Editing is stable and heritable. That means from one generation to another generation can be possible. So gene edited crop, farmers can easily make the seed, not like the, hybrid. Transgene free plants in few generations. So first time maybe, CRISPR gene may present. So within few generations, they are aborted from the DNA, and you can find completely transgene free genome edited plant. As a result, they are safe to use in the environment. Edited plant or variety can be used for plant breeding if new traits are added or new biodiversity is generated. New set of CRISPR-Cas systems such as CPF1, it is also called Cas12a, Cas13a, new enzymes are now discovered, and CRISPR is becoming like Photoshop or user-friendly technology like your mobile phone. And I can foresee that CRISPR would be the main vehicle or tool or technology or toolkit for crop breeding in near future. That you can see in every biology lab, they will use this bioediting tool. So this is all for today, and now it is your time to ask some question. So unmute yourself and ask some question on CRISPR-Cas gene editing. Erin Agnila Rahman. Any question?
Sir, what is sgRNA?
Okay. So any question? Erin Agnila.
Sir, I asked what does sgRNA mean?
Oh, guide RNA. Guide RNA means you design an RNA which is complementary to the your targeted DNA sequence in the genome. So that guide RNA guide the CRISPR-Cas complex system to go and find that sequence, because in case of CRISPR-Cas, you must know the sequence of the certain gene or in the DNA segment which you want to edit. So if you know the DNA sequence, you can make the RNA sequence, complementary RNA sequence, and it is called guide RNA. So guide RNA easily we can synthesize, and there is a software if you know the DNA sequence. So guide RNA is gRNA. Clear?
Thank you, sir.
Welcome. Any other question from any other student?
So,
So who are asking question? More, right?
sir, is gene marker is somehow related with gene editing?
Gene marker.
Gene marker is somehow related with gene editing, CRISPR-Cas.
Gene editing is not related to the marker. Here, you know, marker we use, you know, to amplify specific fragment of the DNA, and by using PCR, we can amplify it and visualize it and follow a specific segment of DNA. But in case of gene editing, we use the guide RNA. Depending on the sequence you want to edit, and guide RNA with the CRISPR-Cas system, you incubate in the cell which you want, where genome you want to edit, and guide RNA guide the system and find the specific location that you are targeted, which is complementary to the guide RNA sequence. And then Cas enzyme cut the make the cut into the double strand, that is double strand break, and the principle I showed you, there are two ways. One was homologous end joining, another one is you know, the recombination. So you can make editing in if several guide RNA you introduce there, you can do multiplex editing, several places at a time, or you can do single gene editing or silencing. Even a single base editing can be possible now. Any question?
Thank you, sir.
Welcome.
So, what's the difference in between CRISPR-Cas9 and Cas12?
CRISPR-Cas9 and 12.
Yes, sir.
Actually 12A or 13A, they are doing the same job. They are the protein or enzyme. They cut the double strand associated with the CRISPR sequence. So job is same, but you know, structurally in case of protein, both can cut the DNA, and 3D structure, they are a little bit structurally different. Scientists discovered from different microorganisms, and it has been found that Cas9 was very popular, but now Cas12 and Cas13, they are found some additional you know, advantages and more efficient, less you know, non-target at editing or like that. So they are the, like you know, your Nokia phone or iPhone or like that. Purpose is same. They are structurally different protein, both CRISPR associated protein or enzyme. And if you want to know the 3D structure of difference between Cas9, Cas12, Cas12A, Cas13A, you can find in the internet, even in the YouTube. There might be some video showing animation where they are different at 3D structure. Any other question?
during editing, some nucleotide present. Is they create any harmful effect?
During editing, some
Nucleotide present. Nucleotide remain. You say
nucleotides are
remain. Is they create any harmful effect for gene editing?
okay. When you target a specific gene to edit, for example, you want to make it non-functional. For example, some of the genes are harmful. They collaborate to the pathogen for the infection. So you cut few nucleotides from that gene, and due to few nucleotides absent in the gene cannot express properly. As a result, you can gene product cannot be shown, and disease pathogen cannot find the host and successfully infect it. So it is not the whole gene is 100% deleted, but it is partly knockout. Lots of nucleotides are remain, but they are not creating problem, because in our DNA or DNA of any organism, huge nucleotides or sequences are non-functional, no expression, or they are not containing the gene or codes for a particular amino acid synthesis. But sometimes if you delete a gene or make a non-functional of a gene, it may have a pleiotropic effect. That means it may affect the expression of neighboring other gene. In that case, it is a problematic. For example, you want to make a disease-resistant crop. You deleted a disease-susceptible gene, S gene. It may affect the gene associated with the growth and development of the that plant, and then yield reduction may be taken place. So after the gene editing, it is not the final. You have to see the phenotype. What happened in the phenotypic properties. That means the other properties, agronomic properties and features of the plant. It is very important. So we cannot give 100% guarantee that the gene edited in the laboratory would 100% show the desired effect in the field. Maybe desired effect in case of plant disease resistance, but it may have undesirable effect with expression on expression of the other valuable and useful genes. Clear?
Sir, thank you.
Then thank you very much. If you have no question? No question? Okay, excellent. Thank you very much, and I wish you a safe and healthy stay.